Howdy, folks. Thanks for checking into Mr. Ulrich's landofbiology.com. I am Mr. Ulrich. In this notes cast, we're going to be taking our first look at Gregor Mendel, a mid-19th century Austrian monk who spent a lot of time breeding peas and taking notes on those peas. And it was from these notes that he was able to work out the basic rules of inheritance, how traits are handed down from one generation to the next, even though he had no idea what DNA was and the word gene didn't even really exist. In this first part, we're going to take a look at Gregor Mendel and how he did his work. And in part two, we'll start to look at some of Mendel's laws and how those work. Sure, modern genetics began in the mid-19th century in an Austrian monastery, but we can look even further back. The Bible has stories about genetics. Jacob and Laban, and you can look up the specifics back in Genesis, but to paraphrase a much longer story, Jacob agreed to take care of Laban's goats for the price of some of the goats. He would keep some of the goats that had a particular trait. Unbeknownst to Laban, what he did is he separated the goats, and through some other kind of scientifically suspect means, made sure that most of the goats had the trait that would give them to Jacob, and so he ended up with most of the goats. Now, Gregor Mendel did something a little bit more scientific. He employed quantitative analysis through rigorous note-taking of cross-pollination amongst peas and keeping very careful track and counting up the characteristics of the offspring in order to figure out the basic underpinnings of the laws of inheritance. Mendel's work with pea plants relied on careful manipulation of the process of pollination. So he would cut the male parts off of the some flowers before they would grow to term to keep them from generating pollen. He would bag the female parts of other flowers to keep them from being pollinated. And then he could control which pollen grains were fertilizing which female flowers by actually painting the pollen grains onto the pistils. And by doing this, he would generate a group of flowers of pea plants that would all have, say, white flowers. Whenever they cross-pollinated with each other, they always had white offspring, white flowering offspring. He would do the same with purple flowering offspring, and so that all of their offspring were purple flowering. And he called these the true breeding parents, or the pea generation, the parental generation. He would then carefully manipulated so that he would crossbreed those and raise the seeds and observe the traits in this next generation, which he called the F1 or first filial generation. He would allow those to self-pollinate, so he would bag each one of those. And so they would self-pollinate, and the next generation would be called the F2 generation. Flower color was only one of several different traits that Mendel looked at. He also looked at the color of the seeds, the shape of the seeds, the shape of the seed pod, where the flowers were located, and even the height of the plants. As he followed these traits from one generation to another, he noticed a pattern in his notes. When he took parents of two different traits, all of the F1 generation would have one of either parent's characteristics or phenotype. When they were allowed to self-pollinate, he always got pretty close to the same ratio of phenotypes between the two different P generation parents. It was always a 3-to-1. There was more of the ones that were present in the F1 generation, but there were a few. So that trait of, in this case, white floweredness skipped generations. So it was present in the parent generation, not present in the F1 generation, and then present again, albeit only 25%, but still present in that F2 generation. Through following the patterns of the inheritance of these traits, Mendel was able to figure out that what happens is each parent donates what he calls a factor. We now know them as genes, but each parent donates a factor to the expression of a particular characteristic, say flower color. And that factor can, in the case of peas, be either for a purple flower color or a white flower color. We call these different factors that code for the same characteristic alleles. Now, to put a little more modern spin on this, we know that these genes are organized onto chromosomes. At a particular locus on a chromosome is going to be a particular gene for a particular characteristic. And now we know that there are variations. There can be, in this case, either a purple allele or a white allele at that particular locus. This is the nature of homology. So this is what we mean by homologous pairs of chromosomes code for the same characteristics, though they may code for different traits because they might have different alleles on them. So they code for flower color or flower shape or pod shape or flower location or height in the case of peas, though they might code for different heights or different flower colors or different flower locations. Traits, therefore, are inherited as discrete units. You get one from one parent and one from the other parent, two alleles, and that's where the nature of diploidy comes from. Die for two, one from one parent and one from the other. It's kind of like having two editions of a particular textbook. Like you might have two biology textbooks. They're going to talk about the same stuff, but they're going to have different phrases, different words, different sentences that describe the same concepts. Back in Regents biology, we talked about the law of dominance, that when there are two different alleles present, one allele is going to mask the expression of the other allele. In the case of our purple and white flowers, that we don't end up with light purple when purple and white are crossed. We actually end up with all purple flowers, that the white allele is totally masked. We know now that that dominant allele actually codes for a protein that does something, that actually has a function. And the recessive allele is a gene that's broken. It might not even code for a protein at all. It might be so broken that it doesn't have the right sequence of nucleotides to even generate a protein. We often talk about the dominant alleles as the wild type, as most often alleles are there to code for something. And the mutants are the ones that have something wrong with the allele so that the protein doesn't work. And the resulting trait is the result of not having that protein. Well, now I've used the word phenotype a couple times, and we should probably define it here. The phenotype is how the organism looks. It's the expression of the genes that they have. This is the physical, the pH. That's what the phenotype comes from. The genotype talks about the actual genes that are there. So when we're talking about dominant versus recessive, that's where we're really talking about the genotype. When we start talking about crosses, we typically use capital letters and lowercase letters to show the dominant and the recessive alleles. We got to be careful, make sure that you're using dimorphic letters. P is probably not a real good one. When I print them out here in text form here, you can tell the difference between an uppercase and a lowercase. But when you're writing them on a piece of paper, it's good to choose something like a Q or something like that. That's really different between the upper and lower cases. So here in our purple flowering pea plants, we're going to use the uppercase P to show the dominant purple allele and the lowercase p to show the recessive white allele. So our true breeding purple flowering pea plants are going to have two dominant alleles, and the true breeding white flowering pea plants are only going to have recessive alleles. When we cross the two, we get the big peas crossed by the little peas, all of the offspring, the only resulting possibility, since the dominant purple flowers are going to make gametes that have the dominant allele, and the recessive white flowers are going to make gametes that have the recessive white allele, all of the offspring are going to have one dominant allele and one recessive allele. To apply these terms to the classic Mendelian test cross, here's my true breeding purple flower plants and my true breeding white flower plants. Those are phenotypes because they're physical expressions. To talk about their genotypes, the purple ones are going to have two dominant alleles, and the white ones are going to have two recessive alleles, both homozygous. And all of the offspring are going to be heterozygous, one dominant, one recessive allele. When we let those self-pollinate, that's where things are going to start to get interesting. This is where Punnet squares come into play. Now, what we're doing with a Punnett square is we're actually making a matrix of possible gametic combinations. Since when the plants are going to go through meiosis, those homologous pairs of chromosomes are going to segregate, and each gamete is going to get one of each homologous pair. That means that each gamete is going to get one of each allele. So we're really looking at which alleles each gamete is going to get. So in the case over here of the male, it's either going to get the dominant allele or the recessive allele. And in the female eggs, they're all going to either get the dominant allele or the recessive allele. It's not very exciting, but there's a little variability here. And now it's just a matter of which alleles, which gametes are going to meet to fertilize. And since we assume there's an equal chance of either type of gamete being produced and either type of gamete doing the fertilization deed, we can just go through and do a 4-by here and see that we have a 25% chance of big P, or homozygous dominant. I have in this box a big P and a little p, but I also have another box with a big P and a little p. These are both heterozygotes. And then I have 1/4 of them are going to be homozygous recessive, 2 little recessive alleles. So here's where we get that phenotypic ratio of 3:1. These 3 are going to be phenotypically dominant. They're going to show purple flowers. Even those heterozygotes, they're going to mask that white allele. And the 1/4 of the offspring that have 2 recessive alleles, those are the only ones that are going to have the recessive white trait. So this gives us our 3:1 ratio in the F2 generation. So our genotypes, 25% homozygous dominant, 50% heterozygous gives us 75%. And phenotypically, we get that 3:1 ratio. Well, I've been using these terms, homozygous and heterozygous. We should get them into our notes. Homo is for the same. So when we're talking about homozygous, the zygote is the zygous part. So those homologous pairs of chromosomes have recombined. And at this locus, they're the same. So homozygous dominant, so both dominant alleles, or homozygous recessive for both recessive alleles. We also have the heterozygous individuals, hetero for different, and that's where there are different alleles for that location. The problem for us geneticists is that organisms can have 2 different genotypes for the same phenotype. So how are we going to figure this out? We'll do a test cross. A test cross is when we take that phenotypically dominant individual, we don't know what its genotype is, and we cross that with a phenotypically recessive individual since we know what their genotype is. The test cross is going to work by basically showing us which of the 2 possible outcomes took place. Which ratios did we end up getting? If we end up getting all of the offspring purple, then we know that it was homozygous dominant. If we get actually half of the offspring being phenotypically white, then we know that we're talking about a heterozygote. Well, I think that's enough for part 1. Once again, thanks for checking in. And if you have any questions, feel free to drop me an email or put a question in the comments section or go to Google Classroom or any of that fun stuff. Other than that, we'll see you in class.